to today's video lecture. In today's video lecture, I'll be solving previous year's NEET questions and discussing the answer. As you can see on the screen, it is for the chapter Human Reproduction, and we are in part 2 of the series. So if you haven't checked out part 1, please go and do so. Alright, so let's begin. Okay, so question number 1. Which of the following cells during gametogenesis is normally diploid? Okay, and this appeared in 2015. So let's go through our options. A. Spermatogonia. B. Secondary polar body. C. Primary polar body. And D. Spermatid. Okay, so sorry. Which of the following cells during gametogenesis are normally diploid? Okay, that means 2N. Okay, they have 46 chromosomes in them. Okay. So A, spermatogonia, secondary polar body, no. Primary polar body also no. D, spermatid, no. So it's clearly spermatogonia. Alright, so your answer is A. Let's go to the discussion. Okay, so I thought that, instead of just directly discussing the answer that is there, I thought I'll just, explain spermatogenesis to you so that you understand the whole concept here itself, right? Previously, I've explained oogenesis in part 1, so you can check that out. So today I'll be explaining spermatogenesis. Okay, so spermatogenesis. So in fetal stage, let's see what happens at the fetal stage. Okay, so primordial germ cells migrate into the testes and become spermatogonia. Okay, so that's how spermatogonia appears. Now, spermatogonia are actually immature germ cells. Okay, so you have spermatogonia over here. Okay, so you have spermatogonia here, spermatogonia, and they already formed at the fetal stage from primordial germ cells. The spermatogonia lie in the inner surfaces of the seminiferous tubules. Okay, so you can see that over here. So that was already started at the fetal stage. Now what happens at puberty? So spermatogonia remain there as it is. Now when the male attains puberty, with the release of gonadotropin releasing hormones, obviously, so GnRH is released from the hypothalamus and it acts on the anterior pituitary gland of the male. Okay, and from there FSH and LH. Now LH is in females, it is a similar function in males too, and it's called as ICSH. Okay, so if you see ICSH, they are actually talking about the complement LH hormone in males. Okay, and in turn release hormones from the anterior pituitary gland. Correct? Now what happens? The spermatogonia migrate among the Sertoli cells and proliferate rapidly via mitosis. So when these anterior pituitary hormones act on the seminiferous tubule, the spermatogonia, those are the immature germ cells, okay, they begin to migrate amongst the Sertoli cells. Now Sertoli cells are very, very important. Okay, they are the nurse cells. So they provide nutrition to the cells, the growing male cells. Also, like I've previously explained in the video lecture in part 1, that these male germ cells, they form cytoplasmic bridges with the Sertoli cells. So if Sertoli cells were absent for whatever reason, it would be very difficult. It actually, in that case, these germ cells will not be able to grow into sperms. Okay, develop into sperms if Sertoli cells were not available. So the spermatogonia migrate among the Sertoli cells and proliferate rapidly via mitosis. So they increase in number via mitosis. They become enlarged to form primary spermatocytes. So this is how primary spermatocytes are formed. Okay, so these spermatogonia, they first proliferate via mitosis and they grow in size to form primary spermatocytes. Now each primary spermatocyte undergoes meiotic division. Now please note, like you can see here, this is taken from NCERT, this diagram here. Okay, so you should be familiar with this, right? See, spermatogonia stage, this answers our question also. See, spermatogonia stage, it is 46. Okay, so if n is equal to 23, 2n would be 46. So there are 46 chromosomes per cell. Okay. In primary spermatocytes also, still it is n is equal to 20. That means 46 chromosomes are still present here. So it is still 2n over here, 2n over here, 2n over here. Now, after the first meiotic division, okay, after the first meiotic division, secondary spermatocytes are formed. Okay, that's meiosis 1. Okay, I'll just write it as M1 over here. It says here first meiotic division. Secondary spermatocytes are formed and the chromosome number becomes half. Okay, chromosome number becomes half. That means it becomes 23 now. Okay. Now, after a few days, the secondary spermatocytes undergo another meiotic division to form spermatids. Now, please learn the difference between mitosis and meiosis. It is very important. It is there in cell biology, and because that will then help you understand this and how those chromosomes numbers become from 46 to 23. Right? So secondary spermatocytes undergo another meiotic division to form spermatids. Okay, so spermatids also in the 23 chromosome number is there per cell. Okay, now these spermatids, they convert to spermatozoa by the process of spermiogenesis. Okay, so where is it? Sometimes the pen disappears. I never know where it is. Okay, so spermatids are formed and they differentiate into spermatozoa, and that is with the help of FSH via the process of spermiogenesis. So FSH is a very important hormone which helps convert spermatids to spermatozoa, or the mature sperms that we see, the sperms that are there. Yeah, so FSH, like I said, is very important. So if FSH was absent, then spermiogenesis wouldn't happen. Okay, so FSH plays a very important role here. You can note this down in your notebooks. Okay, this is an important piece of information. Okay, so this is how spermatozoa are formed. Okay, from the spermatogonia. Now if we go back to our question, which of the following cells during gametogenesis is normally diploid? Now, secondary polar body, primary polar, definitely not. Spermatid, no, they are not diploid, become haploid. So n is equal to 23 over here. Okay, they remain as n. The chromosome number becomes half, and we need that. So meiosis 1 and meiosis 2 participate in the formation of germ cells. So when fertilization happens, there's a male cell, sex cell, and a female sex cell. They combine and they form 46 chromosome number, which eventually forms into a human being, right? So for the formation, spermatogonia is 46 chromosome, which is equal to 2n. It's known as diploid cell. Okay. So this was pretty simple. Spermatogenesis is pretty simple as compared to oogenesis. Oogenesis, there are lots of things happening at the fetal stage. There are lots of things happening, and then after that also, because of the menstrual cycle gets involved at puberty for females, and the follicle development happens, and all that is there. I've explained that in part 1. But in spermatogenesis, it's pretty simple. It's very straightforward. It's not very complicated like men. Bad joke. Okay. Let's move on to the next question. Okay, question number 2. What is the correct sequence of sperm formation? It's appeared in NEET 2013. So we define now. I've already explained spermatogenesis. We can see from the options. Yeah, they're all the terms related to sperm formation. So A, spermatogonia. Yes, we start with spermatogonia. Then we have spermatozoa, spermatocytes, spermatids. B, spermatogonia, spermatocytes, spermatids, spermatozoa. Spermatids, no, it definitely doesn't start there. Spermatogonia, spermatocytes, spermatozoa. No, that's incorrect. If you go to the first one, spermatogonia, no, that's incorrect. So it is B. Okay, spermatogoa, spermatocytes, spermatids, and then finally spermatozoa. Okay, so answer is B. I've already explained this in question number 1, right? So now it's very easy for you to answer this question. You already know what spermatogenesis is. Okay, spermatogonia are immature germ cells which enlarge to form primary spermatocytes. Okay, and then primary spermatocytes undergo meiosis 1 to form secondary spermatocytes, which then undergo meiosis 2 to form spermatids. Okay, so this was pretty simple, very straightforward. You had to know spermatogenesis um, very well. Okay, so this is important, just like oogenesis. All right, so I guess we can move on. Oh yeah, spermatids form spermatozoa via the process of spermiogenesis, right? Okay. Question number 3. The figure shows a section of human ovary. Select the option which gives the correct identification of either A or B with function or characteristic. Sapin Karnataka NEET 2013. Okay, so you have A and you have B here. We can see B is a corpus luteum actually. And this diagram is taken from NCERT, so you need to know that. And A is actually primary follicle. Okay. So you have A, B, corpus luteum. Yes. Secretes progesterone. Yes. A, tertiary follicle, forms graphene follicle, but A is not tertiary follicle. B, corpus luteum secretes estrogen. Yes, it does. Corpus luteum also secretes estrogen, but progesterone is more, okay, as compared to estrogen. A, primary oocyte, it is in the prophase 1 of the meiotic division. Now, it's not really primary oocyte because it is showing the whole structure. So, it's actually primary follicle. Okay. So, yeah, A will not be accurate, and just if it's there in the NCERT, just follow what's written in NCERT. Don't spend too much time in analyzing it with respect to the exam. So your answer is A. Okay, it's corpus luteum and secretes progesterone. That's the appropriate answer amongst the options that are given below. Okay, so you have the same structure here. So you see this corpus luteum here, and you have a primary follicle over here. A is primary follicle, B is corpus luteum. The granulosa cells of corpus luteum secrete progesterone and estrogen. Like I said previously, it is mostly progesterone, but also secretes some amount of estrogen also. Okay. So clear? Let's move on then. Like I said, NCERT diagrams are very, very important. Okay, so please study those diagrams. Like you got that diagram in the question over here. So if you knew the diagram, it would be very easy for you to answer this question. Okay, let's move on. Question number 4. Which one of the following statements is false in respect of viability of mammalian sperm? This appeared in 2012. So we are talking about viability. Now viability means what? Whether a sperm is dead or alive, right? So A, sperm is viable, and we have to find the false statement here. A, sperm is viable for only up to 24 hours. Okay. B, survival of sperm depends on the pH of the medium and is more active in alkaline medium. Okay. C, viability of sperm is determined by its motility. Okay. D, sperm must be concentrated in a thick suspension. Okay, so which one is false here? A, sperm is viable for only up to 24 hours. Yes. Survival of sperm depends on the pH of the medium as more active in alkaline medium. Yes, that's correct. Now C, viability of sperm is determined by its motility. No, that's not the criteria for determining whether a sperm is viable or not. Just because it is motile, yes, it's alive, but just because if it's immotile doesn't mean it's alive or dead. We don't know that as yet till we do further tests on it, or do further analysis on it. Okay, if something is not moving doesn't mean that it's dead, right? So motility is not a criteria to decide whether a sperm is dead or alive. Okay, so C is the right answer. And of course, D, sperms must be concentrated in a thick suspension. Okay, so if if the semen is diluted, the sperms don't survive for long. Okay, they start aging. Okay, and eventually die. Okay, so which is false? C is false. Answer is C. Now, this is a very good question. It required a lot of research to be done with respect to understanding the viability of a sperm. So yeah, this deserved a smiley face because it was more of an application type of question. You had to really like put in your mind into this. Yeah, so sperm can live for many weeks in the suppressed state in the ducts of the testes. Okay, so when they are in the suppressed state in the testes before ejaculation, they can remain there for many weeks. Okay, they are alive there for many weeks. But the ejaculated sperm, now once the semen is ejaculated into the vaginal tract, they can only live up to 1 to 2 days, that is 24 to 48 hours. Okay, the majority of the sperm that is ejaculated can only live up to 24 to 48 hours. Okay, so you can expect the life expectancy of a sperm to be around that much, okay, that many days in the female genital tract. Now, only few sperms remain alive for 5 days in the female genital tract. Okay, so don't take 5 days as all the sperms are alive for 5 days. It is not true. The majority of the sperms are alive for only 1 to 2 days, but only a few sperms remain alive for only 5 days. So it is a way to determine how, if a woman wants to get pregnant, or to determine fertility, how and when you can plan fertilization, right? That way this information is useful, that only few sperms remain alive for 5 days, so a woman can plan based on her ovulation period, right? But if we are thinking about it in terms of sperm viability, then majority of the sperms only remain alive for 1 to 2 days. That's the life expectancy. Okay, it is not 5 days. 5 days is very few sperms only, not the majority of the sperms. So don't go by information that's available on the internet. Please refer to proper books or speak to some teacher regarding this answer, because I know many people go and Google here and there and they find incorrect answers, but those are incorrect answers, so don't fall for that. Okay, now sperms remain viable in surrounding alkaline conditions, which is true. Okay, that is around pH 6 to 6.5. Acidic conditions are not favorable for sperms to remain alive. Okay, the sperms need alkaline condition. The prostate gland secretions maintain that to counteract the acidic secretions in vas deferens and in the vaginal secretion. Now, in vas deferens, there's some citric acid present, and there are some metabolites of the sperm formation that are present there, which make it acidic. And also the vaginal tract of the female is acidic by nature, okay, which is around pH 3.5 to 4. Okay, so now the prostate gland secretions maintain that alkalinity, okay, because it needs the sperms to live, right? So to maintain that, prostate gland has secretions which counteract the acidic secretions. Okay, so yeah, the sperms remain, yes, it's true that the sperms remain viable. Okay, they are alive in surrounding alkaline condition, which is around pH 6 to 6.5. Okay, now viability of sperm is determined by assessing the membrane integrity of the cells. Okay, now first, if somebody is checking or doing a semen analysis and they see motility, yes, the motility is there, that means the sperm is alive. But there will be cases where the sperm won't be motile, it's immotile, but that doesn't mean it's dead. Okay, it might still be viable. So motility is not a way to determine whether sperm is viable or not. Okay, so what they do is they assess the membrane integrity. So remember that there's a plasma membrane around that. So they check whether that membrane integrity is still intact. It's still integral part of the spermatozoon and it has not been disrupted. Okay, in any way. Now, how do they do that? Now, this is done by staining the cells with a dye. And if the cells are damaged, the dye is taken up by the cells. Okay, so they do test in the lab where they stain the sperm cells. Okay, and if the cells take up that dye, means it is damaged, and if they don't, that means the membrane is still integral. Okay, it is still intact. So that is one way. The second test is hypoosmotic swelling. Okay, so live cells when placed in a hypotonic solution will swell and dead cells will not. So if a cell is alive and you place them in a hypotonic solution, they will swell up. This is osmosis, right? But if it's dead, then nothing will happen. The cells will not swell up like they should. So this is the second way to check whether sperm cells are dead or alive. Okay, so these are the two ways by which semen analysis is done with respect to whether the sperm is viable or not. Okay. All right. So the fourth point was whether sperms are put into or are concentrated in a thick suspension, right? So sperms are suspended in semen, which is around pH 7.5, giving it a milky white appearance from prostatic gland and a mucoid consistency from the seminal vesicles and mucus glands. We know that, right? We know that the semen as such is thick in nature, right? And the sperms are present inside that, okay, because of secretions from the seminal vesicles, from the prostatic glands, from the Cowper's glands, all that. There's a mix of everything. Now, studies have found that with increasing dilution of semen, there is a loss in sperm viability. Okay. The reason, now this is one of the reasons. Okay, now I had to do some research on this particular topic on how thick concentrated semen plays a role in viability of sperms. So this is one of the reasons by which researchers have come to the conclusion that sperms remain viable. The reason could be that in a thick concentrated suspension, sperms are able to conserve energy. Okay, this is a reason why sperms remain alive in a thick concentrated suspension. Okay, they don't move, there's no space to move around much, so they are able to remain together. Okay, and they are able to conserve that energy, so which keeps them alive, okay, in that thick concentrated suspension. Now, with dilution, it has been found that there is a gradual decay or exhaustion of food reserves leading to aging of sperms. Okay, so as the semen was diluted, they found that there's a gradual decay or exhaustion of those sperm. There's an aging of sperms. It could be that there is a gradual decay or exhaustion of food reserves. So as and when if something is moving around, it is using up energy, right? So if it keeps moving around, the space provided to them, they'll move around. And so they begin to age. The food reserves are consumed or exhausted and they begin to age. Now this is based on research that has been found and which I thought was applicable to answer this question. So this particular question took a lot of time for me to come to a conclusion, and I found this question pretty good because it did require you to think about it. So yeah, please don't fall for Google random researches. Okay, so sperm is viable for only up to 24 hours. Yes, that is true. Okay, but it does not apply to this question because we have to find the false statement here. So here it's wrong. Survival of sperm depends on the pH of the medium. Yes, and is more active in alkaline medium. Yes, that's true. Okay, and viability of sperm is determined by its motility. No, that's incorrect. There are other tests done that determine whether a sperm is viable or not. Okay, just because the sperm is not moving doesn't mean it's dead. Okay, so that is not a criteria to determine whether a sperm is viable or not. And the test that I mentioned regarding viability of sperm was taken from WHO semen analysis. So they have a manual to tell you how semen analysis is done. And yeah, sperms must be concentrated in a thick suspension. Yes, I have already explained that to you. That is true, but in this case we have to find the false statement, so C is the false statement. Okay, I guess we can move on then. If you like, you can note this down in your notebooks. It'll be helpful. Okay, so question number 5. Which one of the following statements about human sperm is correct? Okay, and this appeared in 2010. A. Acrosome has a conical pointed structure used for piercing and penetrating the egg, resulting in fertilization. Okay. The B. The sperm license in the acrosome dissolve the egg envelope, facilitating fertilization. Okay. C. Acrosome serves as a sensory structure leading the sperm towards the ovum. Okay. And D. Acrosome serves no particular function. Okay. Now, the acrosome, they're all talking about the acrosome A, B, C, and D. But the acrosome is a conical pointed structure, but it is not really there for piercing the egg, right? That is incorrect. But it contains a lot of enzymes, proteolytic enzymes. So the correct answer is B. Sperm license in the acrosome dissolve the egg envelope, facilitating fertilization so that they can then penetrate the egg. Okay. So the answer is B. That's the correct statement. Okay. So acrosome contains large quantities of hyaluronidase and proteolytic enzymes. Okay, so hyaluronidase creates a pathway towards the zona pellucida of the ovum via the granulosa cells. So if you look at the structure, there's a zona pellucida, which is a glycoprotein. There are some granulosa cells, so corona radiata. So the sperm has to first penetrate through the granulosa cells, go via the zona pellucida. Correct? So for that, there are some enzymes present. Okay. There are some enzymes present in the acrosome which then allow this penetration to take place. Okay. So once it reaches the zona pellucida, the acrosome binds with the receptors on the zona pellucida and release the acrosomal enzymes. Okay. So once it has crossed the granulosa cells, it will reach the zona pellucida. Zona pellucida is an acellular layer. Okay, it is made of glycoprotein and there are some receptors present on it. So it will bind onto that and it will release the acrosomal enzymes. So now these enzymes create a pathway through the zona pellucida for the passage of the head into the inside of the ovum. That is when meiosis 2 is completed to form a mature ovum, right? So yes, this is the function of acrosome in a sperm. Okay, I think I've already explained this also before, but okay, I'll explain one more time, no problem. And to answer your question, yeah, the other options were clearly not making sense. So B is the appropriate answer. Okay, so sperm license are present, the acrosome will dissolve the egg envelope, facilitating fertilization, which is correct. Okay, let's move on. Alright, so question number 6. The correct sequence of spermatogenetic stages leading to the formation of sperms in a mature human testes is, this appeared in 2009. So correct sequence of, so I think we've already come across a similar question in the beginning, right? A, spermatogonia. Yes, it starts with spermatogonia. Then spermatocytes. Yes, spermatid and then sperms. Makes sense. B, spermatid. No, it doesn't begin there. Spermatogonia, spermatids. No. So B and C are incorrect. Spermatocyte, no, it doesn't begin there either. So to form sperm, it starts with spermatogonia, then spermatocytes, and spermatid, and then sperms. Right? So your answer is A. Again, you can go back to the beginning of this lecture. I've already explained spermatogenesis to you, so you can refer to that. Now see, if you knew that, then it's very easy to answer this question, right? So correct sequence of spermatogenesis is what you can see here. All right, I will not go into further details. This is a very straightforward question, right? Let's move on. Okay, so question number 7. In humans, at the end of the first meiotic division, the male germ cells differentiate into this appeared in 2008. So end of the first meiotic division, all right, the sperm gels differentiate into A, spermatids, B, spermatogonia, C, primary spermatocytes, and D, secondary spermatocytes. So after the first meiotic division, the male germs differentiate into or form secondary spermatocytes. Okay, so your answer is D. Again, this is again related to spermatogenesis. Oh yeah, so pretty straightforward. Again, this is again related to spermatogenesis. You have to know that thoroughly. Okay, no excuses. Okay, next question then. Okay, so question number 8. Sertoli cells are regulated by the pituitary hormone known as, and this appeared in 2006, A, LH, B, FSH, C, GH, and D, prolactin. So Sertoli cells already discussed are nurse cells, okay, and they are regulated by B, FSH, okay. FSH plays an important role in spermatogenesis. Answer is B. So there are FSH receptors present on the Sertoli cells to which FSH binds to, right? So it allows the Sertoli cells to grow and secrete spermatogenic substances which help in spermatogenesis along with testosterone from Leydig cells. Now, Leydig cells behave or regulated under the influence of LH, which then secrete testosterone. So, FSH or Sertoli cells alone don't play a role in spermatogenesis. They also need testosterone to help in spermatogenesis. Okay. So, but FSH receptors are present in Sertoli cells, which then secrete spermatogenic substances, which then help in spermatogenesis, and that obviously is along with testosterone. Oh, sorry. Also, FSH plays a role in spermiogenesis. Okay, so basically the whole spermatogenesis, FSH is doing an important role. But also spermiogenesis, FSH also plays an important role. Okay, that is a differentiation of spermatids into sperms. Right, so clear, this was also direct. We'll move on. Okay, so question number 9. The middle piece of the sperm contains, and this appeared in 99, A, proteins, B, mitochondria, C, centriole, and D, nucleus. Very important question. Okay, so you need to remember the structure of sperm. Okay, so middle piece of the sperm contains B, mitochondria. Okay, answer is B. Yeah, so here I'm sure if you watched my previous lecture, you'll be familiar with this structure. So you can see the mitochondrial sheath around the axial filament, this pink structure here. Okay, and in spermiogenesis, that means when spermatids are converted to spermatozoons, part of the axial filament between the head and the annulus becomes surrounded by mitochondria and together form the midpiece. Okay, so this is the middle piece. So the axial filament between the head and the annulus becomes surrounded by this mitochondrial sheath. Okay, and this forms the midpiece of the sperm. So it is important that you remember the structure of sperms. Okay, what is present in the head, what is present in the neck, and then what the middle piece, and then the tail. You know, how is it all arranged? I've already discussed spermiogenesis in part 1. Okay, so I won't go into this in detail over here, but it is important that you remember. Okay, let's move on then. Okay, so question number 10. How many sperms are formed from a secondary spermatocyte? Okay, and this appeared in 1990. A4, B8, C2 and D1. So we're talking about a secondary spermatocyte. That means one secondary spermatocyte, how many sperms are formed. If you remember spermatogenesis, the answer is 2 here. Okay, two sperms are formed from one secondary spermatocyte. So your answer is C. Let's go to the discussion. So here, yeah, from one secondary spermatocyte forms two spermatids via meiosis 2, and then those spermatids then differentiate into spermatozoa. Okay, so you have one secondary spermatocyte, they form two spermatids and then eventually form the sperm over here. Okay, it is important that you remember this spermatogenesis, from how many cells are forming, okay, via meiosis or mitosis or whatever, you know, where mitosis is happening, where meiosis is happening, and how is the chromosome number changing. Okay, so this is important. The question said from one secondary spermatocyte. Okay, so there are two spermatids formed from one secondary spermatocyte, which then differentiate into spermatozoa. That answers our question. How many sperms are formed from a secondary spermatocytes? Yeah, the answer is C, 2. All right, let's move on. Okay, question number 11. Egg is liberated from ovary in, this appeared in 1989, pretty old question. A, secondary oocyte stage, B, primary oocyte stage, C, ogonial stage, D, mature ovum stage. So egg is liberated from ovary, that means ovulation is happening in the secondary oocyte stage. Remember, we discussed this in oogenesis in part 1, when meiosis 1 is completed. Okay, and so secondary oocyte is formed just before ovulation, okay, in the follicle, in the tertiary follicle. So the answer is A. So before ovulation, meiosis 1 is completed to form secondary oocyte. So hence egg is liberated from the ovary at secondary oocyte stage. I have not discussed oogenesis in detail now because I've already discussed this in part 1. So please go check that video out. Okay, so this answers our question. Pretty direct if you knew ovulation, oogenesis well. Okay, let's move on. Question number 12. Which of the following hormone levels will cause release of ovulation from the Graphene follicle? Okay, this appeared in NEET 2020. It's pretty recent. Which of the following hormone levels will cause release of ovum? Okay, so you have A, high concentration of estrogen, B, high concentration of progesterone, C, low concentration of LH, and D, low concentration of FSH. So when ovulation is happening, there is high concentration of progesterone. Okay, there's high concentration of progesterone because 2 days before ovulation there's an LH surge, and I'll explain that in the discussion. So the answer is B. Okay, so you have the diagram here of the ovarian cycle, follicle cycle, right? That's the menstrual cycle. So here what you can see, there's an LH surge here. Yeah, before ovulation, 2 days before. This is 14th day. If your average cycle is 28 days, let's say this is the 14th day. So around the, if not exact, maybe around this blue line is LH. Maybe around the 12th day, let's say 12th day, there is a surge of LH hormone. Because of that, you can see a spike in progesterone. You can see a spike in progesterone, right? There's a surge of LH hormone 2 days before ovulation. You can see the spike in progesterone hormone here. Okay, now this is very important. LH surge is important because it allows many things to happen, which eventually allows ovulation to happen, and I'll discuss that. So what happens is about 2 days before ovulation, there is a surge of LH, which is 6 to 10 fold. So it's a very big surge. It's a big spike. Okay, and FSH also increases, but 2 to 3 fold. You can see that it is not so much as LH. Okay, and both these hormones causes swelling of follicle before ovulation. Okay, so that is the reason why this LH surge and this FSH surge happens. The follicle, ovarian follicle needs to swell, okay, to allow the oocyte to come out of it. Now, LH acts on the granulosa and theca cells and convert them to progesterone secreting cells. Okay, so LH plays an important role in converting those granulosa cells and theca cells into progesterone secreting cells. That's why you see this jump in progesterone. Okay, now secretion of estrogen, or in this case is estradiol. Estrogen is a group of hormones, so estradiol is the one, falls one day before ovulation. Yeah, you can see this drop here. Estradiol is dropping one day. So the dotted line means ovulation. Okay, this is ovulation. Now, one day before that, progesterone LH surges happen, FSH surges happen. Progesterone also begins to increase because LH has now played a role in converting granulosa and theca cells into progesterone secreting cells. Fine. But one day before that, estradiol, estrogen levels also drop. So estrogen or estradiol is not high during ovulation. Actually, there's a drop in estradiol concentration in the blood. Okay, that is at the time of ovulation. Now, this condition result in the release of ovum. That is when ovulation happens. Okay, so these factors are very important. The LH surge, FSH surge, they cause the swelling of the follicle. Because of that LH surge has happened, it has converted the granulosa and theca cells into progesterone secreting cells. At the same time, estradiol has dropped one day before the ovulation, and then these conditions allow the release of the oocyte into the fallopian tube, okay, out of the ovary, and that's what is ovulation. Okay, so if you go back to the question, which of the following hormone levels will cause release of ovum? That's ovulation from the Graphene follicle. In this case, there is no low concentration of LH and FSH, so that is clearly incorrect. High concentration of estrogen, definitely not. There is high concentration of progesterone. You see a spike in progesterone, right? So the correct answer is B. Okay. All right, clear? Yes, we can move on. Okay, so question number 13. No new follicles will develop in the luteal phase of the menstrual cycle because, and this appeared in Orissa NEET 2019. Okay, so no new follicles develop in the luteal phase. Okay, why is that? A, follicles do not remain in the ovary after ovulation. No. B, FSH levels are high in the luteal phase. C, LH levels are high in the luteal phase. D, both FSH and LH levels are low in the luteal phase. Okay, so for new follicles to develop, you need FSH and LH. And if FSH and LH are not present, especially in the luteal phase, they are low. There will be no ovarian follicle development. Look at it logically, right? So your answer is D. Answer is D. Okay, let's go to the discussion. So during the luteal phase, corpus luteum, okay, secrete progesterone and estrogen, mostly progesterone, which has a negative feedback on the anterior pituitary gland. Now, when there is too much of progesterone and estrogen present there, it sends a negative feedback to the pituitary gland in the brain. Okay, that stop, we don't need FSH and LH anymore. Okay, stop the secretion of that. This inhibits the secretion of FSH and LH. Okay, and so during that time, there's also secretion of inhibin from the lutein cells. Now, inhibin is another hormone that is secreted by the corpus luteum, which inhibits the secretion from anterior pituitary gland, especially FSH. So it plays a very important role in blocking FSH secretion from the anterior pituitary gland. Okay, so the presence of progesterone, presence of estrogen, presence of inhibin, all together play a role in blocking FSH and LH secretion. And FSH and LH both are important for development of new ovarian follicles. So during that phase, ovarian follicle development does not happen during the luteal phase of the cycle. Okay. So to go back to our question, yeah, so no new follicles develop in the luteal phase of the menstrual cycle because both FSH and LH levels are low in the luteal phase. Okay, clear? Yeah. Okay, sorry. The loss of FSH and LH causes degeneration of corpus luteum, which ends 2 days before menstruation. Now, this is explaining how menstruation takes place, how the bleeding happens. Okay. So now when there's no FSH and LH present because of high estrogen progesterone inhibin in the secreted by the corpus luteum, there is no maintenance of corpus luteum. So corpus luteum then slowly begins to degenerate, which is its normal cycle, right? It has to degenerate. So 2 days before menstruation, there is degeneration of corpus luteum. Okay. And before menstruation, there is sudden loss of estrogen, progesterone, and inhibin. Now, if corpus luteum is, it's like a vicious cycle. Okay, you're stuck in a vicious cycle. Now, to maintain corpus luteum, we need LH and FSH, but there is too much of estrogen, progesterone, inhibin in the blood. There is no LH and FSH influence on corpus luteum. So, 2 days before that, before menstruation, it degenerates. Okay, there's involution of corpus luteum, which forms corpus albicans. So when this degeneration happens, what happens? There will be sudden cessation of, there will be a sudden stoppage of secretion of these hormones, estrogen, progesterone, inhibin, right? Now that removes the negative feedback inhibition. Now when there is sudden loss of estrogen, progesterone, and inhibin, menstruation begins. Okay, menstruation begins. Okay, now when these hormones are absent, again then positive feedback loop will start, right? Again LH and FSH will start secreting, right? And so that time ovarian follicle development begins again. So the same cycle repeats itself every month. Okay, so this is how menstruation also happens. So this removes the negative feedback inhibition. So this once again allows the secretion of FSH and LH and formation of new follicles. Okay, so I've explained how corpus luteum plays a role and how menstruation started, how degeneration of corpus luteum then eventually results in menstruation. So basically this is what's happening. All right. Yes, we can move on. Let's see. Go back to the question. Yeah, so both FS and LH levels are low in luteal phase. We've answered the question. Okay, so we can move on. Okay, question number 14. Match the items given in column 1 with those in column 2 and select the correct option given below. It's appeared in NEET 2018. Column 1 has A, proliferative phase, B, secretory phase, and C, menstruation. And then you have column 2. Okay, so let's match. Proliferative phase is follicular phase, so it's A2. Secretory phase is luteal phase, and menstruation is breakdown of endometrial lining. So you have A2, yeah, B3, yes, and C1. So your answer is C. Okay, answer is C. Pretty simple, easy. Proliferative phase is follicular phase, secretory phase is luteal phase, and menstruation is breakdown of endometrial lining. Okay, so this answers our question. Let's move on. It's pretty simple. Okay, so question number 50. Changes in gonadotropin releasing hormone pulse frequency in females is controlled by circulating levels of, and this appeared in NEET 2016. So remember, in females, when gonadotropin releasing hormone is secreted from hypothalamus, it happens in pulses. Okay, there are phases like this. Okay, that's how the GnRH hormone is secreted in the from the hypothalamus in females. Okay, so how is this controlled? A, progesterone only, B, progesterone inhibin, C, estrogen and progesterone, and D, estrogen and inhibin. Okay, so the answer is actually C, progesterone and estrogen. Answer is C. Now, estrogen and progesterone control the pulse secretion of GnRH. Okay. Now, estrogen in small amounts have a strong effect in decreasing FSH and LH, and along with progesterone, which has a little effect on its own, have a multiplied effect. Okay, so progesterone alone does not have a very strong impact, but estrogen has a strong impact. So, but along with progesterone, they have a multiplied effect. Okay, so they are able to decrease LH and FSH. Okay, and also act on hypothalamus. Also, there's actually, if you look at it biologically, it has been said, and it's there in Guyton, I believe, which is a textbook on medical physiology, which has been stated that it has a more effect on anterior pituitary than in the hypothalamus. I'm not saying it does not have, there's no black and white in biology. It's always in between. There's either less effect or more effect, right? Think of it as this way. It's not like zero or full effect, right? So it has a more effect on the anterior pituitary than on the hypothalamus, but that does not mean there is no effect on hypothalamus. Yes, there is a effect on hypothalamus. Both act primarily on anterior pituitary, lesser extent on the hypothalamus by altering the GnRH pulse frequency. Okay, so like I said, don't take it as 0 or 1, that it does not have any effect on hypothalamus. Yes, it does have an effect on hypothalamus, but to a lesser extent as compared to anterior pituitary. Oh, sorry. Inhibin, I just wanted to mention that inhibin also has an effect on the anterior pituitary, but only stops FSH. It does not really have a direct effect on hypothalamus, right? So your answer over here should be C, estrogen and progesterone. That would make sense, right? I guess we can move on. Okay, so question number 16. Select the incorrect statement, and this appeared in NEET 2016. A, LH and FSH decrease gradually during the follicular phase. B, LH triggers secretion of androgens from the Leydig cells. Okay, and C, FSH stimulates the Sertoli cells which help in spermiogenesis. D, LH triggers ovulation in ovary. So D is correct, so which is incorrect here. I mean, not the right answer here. FSH stimulates Sertoli cells which help in spermiogenesis. I've already discussed this. Yes, it's a true statement. LH triggers secretion of androgens from the Leydig cells. Yes, but in males it is ICSH, but it has a similar effect as LH in females, right? So B is correct. LH and FSH decrease gradually during the follicular phase. No, that's incorrect. So A is the incorrect statement here. The answer is A. Okay. Okay, so during the first few days of the ovarian cycle, that's the menstrual cycle, there is an increase in FSH and LH. Okay, there's more of FSH, you can see here, and there's a slight increase in LH. Okay, FSH and LH are important as and when the follicular phase also begins. FSH and LH play a very important role in the ovarian follicle development. Okay, so they definitely don't decrease, gradually decrease. In fact, by the end of the follicular phase, as before ovulation, both of them are going up. Okay, so they don't gradually decrease. In fact, they're gradually increasing. So FSH causes growth of 6 to 12 primary follicles, which is the FSH's job to do, until the growth of the antral follicle. That means when the antrum starts forming in that follicle stage, the physiology is governed by FSH. Okay, so FSH till that stage, FSH is doing the job. Now, when the antral stage is formed, estrogen begins to secrete in that follicular fluid, the antral folliculi. Okay, so now when estrogen is there, FSH is there, they have a very positive effect, okay, on the anterior pituitary gland. What happens is they allow more FSH to secrete. At the same time, they increase the sensitivity of FSH by increasing the receptors on the follicula, in the ovary. Okay, so presence of estrogen secreted into the follicle causes increase in FSH receptors, okay, of granulosa cells. Now this creates a positive feedback, making the granulosa cells sensitive to FSH. The more receptors are there, the more FSH will come and bind to it, right? This also has a further effect. Now, FSH and estrogen, they promote LH receptors. Okay, they promote LH receptors, allowing LH stimulation, creating a more rapid increase in follicular secretion. Okay, now LH, LH hormone is very important in formation of estrogen. Okay, now what happens is when you have, I think I've already explained this in endocrine system, you have the theca cells, TC, I'll just call it TC, and you have the granulosa cells, right? So LH has an effect on both these cells, okay, theca cells and granulosa cells. So let's say LH is bind over here, and what is it, granulosa cells, LH binds over here. Okay, now with the help of cholesterol, progesterone is formed. Progesterone is formed here also. Now what happens is progesterone moves into this, okay, and progesterone converts to androgens. The androgens cannot be used as such. So what happens, androgens moves into the granulosa cells and they convert to estrogen. Okay, this is the synthesis of, you need to understand the synthesis of estrogen. Where is estrogen actually coming from? Okay, so androgens move into the granulosa cells because there is an enzyme present there called aromatase. Okay, and that helps in the conversion of that androgens to estrogen. Okay, which we need. Okay, that's why there is lots of estrogen. Now, LH receptors help in doing that. Okay, so if LH receptors were not there, then it would be very difficult to form more estrogen, and then we need and because of that there's a rapid increase in follicular secretion. Okay, it helps in the maturation of the follicle. Okay, so that's very important part of the follicular phase. Now obviously, now before ovulation occurs, now we know for as a fact that only one follicle is ovulated at one time, okay, in one period cycle or menstrual cycle, right? Only one follicle then ovulates. Okay, so what happens, oh, one ovum, oocyte rather. So one follicle matures, the one that secretes more estrogen. Okay, so the best one is taken out, okay, and the rest degenerate. All right, so what happens is the follicle that secretes more estrogen is selected from that group of 6 to 12. Okay, so at one time 6 to 12 follicles are developing, but only one is selected, the rest degenerate. Okay, now this depresses the FSH secretion slightly. Maybe you can say that over here. Okay, a slight depression is there, okay, in this phase. Okay, now it's preparing the follicle is preparing for that oocyte that will then ovulate out of the ovary. Okay, which will hopefully get fertilized. If not, then it will just come out as your period cycle, right? A menstrual cycle. So in that phase, FSH depresses slightly because maybe there's too much of estrogen that creates a negative feedback. Okay, but that does not last for long, because when ovulation happens, or at least 2 days before ovulation, that's before final oocyte coming out of the ovary, there's an LH surge and an FSH surge, which allow for the obviously the combination of the two allow for the ovarian follicle to swell up and then allow the secondary oocyte to leave the Graphene follicle. Yeah, so 2 days, but 2 days before ovulation, although there's a depression, but 2 days before ovulation, there's a LH surge and FSH also increases, and hence FSH and LH don't decrease gradually during follicular phase. So that was an incorrect statement. So you need FSH and LH during follicular phase, but the decrease in FSH, like I said, is only temporary. That's before ovulation. That is when one follicle that is being selected from the remaining, from the group. Okay. So clear? If we go back to our question, select the incorrect statement. LH and FSH decrease gradually during the follicular phase. No, that's incorrect. It's an incorrect statement. The rest were all correct. Okay, so clear? I guess we can move on. Alright, question number 17. Identify the correct statement on inhibit. This appeared in NEET 2016. A is produced by granulosa cells in ovary and inhibits the secretion of LH. B is produced by nurse cells in testes and inhibits the secretion of LH. C inhibits the secretion of LH, FSH and prolactin. D is produced by granulosa cells in ovary and inhibits the secretion of FSH. Yes, the correct statement is D. I've already discussed this previously. Now see, this was helpful, right? Answer is D. So inhibin in females is secreted by the granulosa cells of the corpus luteum in the ovary, which inhibits the secretion of FSH. Okay, so during the luteal phase, this happens. Obviously, estrogen and progesterone are also playing a role at the same time in inhibiting FSH and LH secretion, but they also have a role in controlling the pulse frequency of GnRH also. But inhibin also plays an important role and indirectly inhibiting the secretion of FSH. Okay, so that is the function of inhibin in females. So the absence of FSH and LH causes degradation of corpus luteum. Oh, sorry. Let's go back to the question. Identify the correct statement on inhibin. It is produced by the granulosa cells in ovary and inhibits the secretion of FSH. Okay, so this is a very important fact to remember. Please note this down if you like. Okay, so question number 18. Which of the following events is not associated with ovulation in human female? This appeared in 2015. So event that is not associated. Okay, so you have A, release of secondary oocyte. Yes, that's associated with ovulation. B, LH surge. Yes, very much. C, decrease in estradiol. Yes, that also happens. D, full development of Graphene follicle. It's not directly associated with ovulation. Ovulation is a different process, right? It's the release of ovum. But Graphene follicle is already formed before that. Tertiary follicle is converted to Graphene follicle. All that maturation is happening before that only, right? So I would say D is the appropriate answer here. So it is an event that is not associated directly with ovulation. So the answer is D. So release of secondary oocyte, LH surge, yes, and decrease in estradiol, yes. There's also decrease in estradiol one day before ovulation, which and it's also helpful that event is also helpful for ovulation to occur. Okay. Full development of Graphene follicle occurs before the ovulation. That is much before that. The Graphene follicle is ready, and even the meiosis one is completed inside. It's in the secondary oocyte stage. So it's not really directly associated with ovulation, the process of release of the ovum, right? But the other events, the release of secondary oocyte, LH surge, and decrease in estradiol, they help in the occurrence of ovulation. I've already discussed this few questions before. You can go back and check my discussion there. I've explained it in more detail. But for this question, this is actually a good question. This was a good question. Okay, it's a good question. It deserved a smiley face. Another smiley face today. So yeah, the full development of Graphene follicle happens before the ovulation. It's already prepared to do its job to release the ovum, the oocyte. But of course, many other factors are happening before that, which then allows for the release of oocyte from Graphene follicle. All right, so yeah, which of the following events is not associated with ovulation? It's D. Okay, I guess we can move on. Question number 19. The main function of mammalian corpus luteum is to produce, this appeared in 2014, A, estrogen only, B, progesterone, C, human chorionic gonadotropin, D, relaxin only. So the main function of corpus luteum is progesterone, also estrogen, but not so much. But A obviously is not the appropriate answer for this. B is the appropriate answer for this. So the answer is B. Okay. So after ovulation, the follicle is converted to corpus luteum with the help of LH. Okay. And the granulosa and theca cells convert to lutein cells. Yes, and secrete progesterone and estrogen, but majorly progesterone. All right. There is some estrogen also being secreted, but mainly it's progesterone. Of course, I've already discussed corpus luteum in endocrine system also in detail, and also in oogenesis also. So I'll not go into details of corpus luteum, right? But this should be easy to understand and to answer if you knew your theory well. Right, so if you go back to the question, yeah, the main function is to produce progesterone. As per the question, yes, B is the right answer. That's the main function. Okay, question number 20. Menstrual flow occurs due to lack of, and this appeared in NEET, 2030. A. Oxytocin. No. B. Vasopressin. No. C. Progesterone. Yes. D. FSH. No. The menstrual flow. That means menstruation, right? Menstruation occurs due to lack of progesterone, right? So that is when after corpus luteum degenerates. So it is C. Answer is C. I've already discussed this also a few questions back. So menstrual flow is due to lack of progesterone, estrogen, inhibin from the lutein cells. Okay, so corpus luteum degenerates, right? So estrogen majorly and progesterone decrease FSH and LH secretion from the corpus luteum. Inhibin decreases FSH secretion. Yeah, so the decrease results in degradation of corpus luteum. So 2 days before menstruation, there is degradation of corpus luteum. Okay, now when corpus luteum degrades, what happens? All these hormones will disappear. Estrogen, progesterone, all that will disappear. So the cessation of progesterone, estrogen, inhibin results in menstruation in the beginning of the next ovarian cycle. I've already discussed this few questions before. It is the same discussion. This is the reason how or why menstruation happens. Okay, that is the bleeding part, the menstrual phase of the menstrual cycle. Okay, I guess that was the menstrual flow. So they're talking about menstruation. That is due to lack of progesterone. Okay, clear. Let's move on. Question number 21. The secretory phase in the human menstrual cycle is also called, and this appeared in Mains 2012. A. Luteal phase and lasts for about 6 days. B. Follicular phase and lasts for about 6 days. C. Luteal phase and lasts for about 13 days. D. Follicular phase and lasts for about 13 days. So secretory phase is also known as luteal phase in the menstrual cycle. So it is called luteal phase and lasts for about 13 days. Okay, and the answer is C. Okay, answer is C. So secretory phase is also known as luteal phase and lasts for about 13 days. So this diagram is very accurate diagram for for the menstrual cycle or the ovarian follicle cycle. Yeah, so ovulation has happened now. So this is the luteal phase. Okay, till the end of the cycle, right? So this is about this. Let's say this is the 14th day. This is the 20th day where menstruation begins. Okay, the next menstruation cycle will begin after this. Menstruation begins to occur. So this will last about 13 to 14 days. Okay, so this is the answer to your question. Luteal phase will last for about 13 days. Okay, clear? Okay, so we've come to this end of this video lecture, and I hope you've enjoyed today's video lecture. I hope you learned something new today. And please don't forget to like, share, and subscribe to MJ NEET Biology. And I'm available on Facebook, Twitter, and Instagram, so you can follow me there for more updates. And yeah, I'll see you in the next video lecture, right? So bye.